Hello everyone, in this video I'm going to go over the test review sheet so you can better understand mutations and karyotyping. The first question is, what is the definition of a mutation? I would call it a change in the sequence of DNA bases, or the change in the sequence of DNA nucleotide. The two causes of a mutation is so the first one would be DNA replication. So during the DNA replication process, right, just like when you're copying anything down, right, even if you were to write an essay and you're copying someone else's essay, you still might make some mistakes. You've done that, I think. But either way, if you're making copies of something, there could be mistakes. So DNA replication could definitely cause change in DNA nucleotide sequence. Right? These are all things that we talk about in class, so there's not really nothing special about it. But okay, the next one is another possibility is transcription. So during the transcription process, DNA is used as a template for RNA, right? So during this process, it's also possible that we can have a change in the sequence of the RNA, which is going to change the protein that's formed using the RNA as a template. So although we say the definition of a mutation is change in the DNA nucleotide sequence, because ultimately that's usually where all the changes start, all the changes of any kind of mutation start in the DNA, but during transcription it's also possible to make mistakes. The other possible cause of a mutation would be the environment. So things such as radiation, right? UV light or UV radiation from the sunlight, or other types of radiations. Smoking, for example, can be cancer causing, or eventually could be cancer causing, but it causes more mutation to happen in your body, more than the normal amount. There could be certain food that you eat that might be, make you more susceptible to changes in your genes. There could be viruses and bacteria that could introduce mutations to your body as well. So if we're talking about mutation, right, DNA eventually is expressed, we call this gene expression. So DNA is expressed into proteins. DNA can't just do work on its own. It has to be used as a template to make an RNA molecule before the RNA molecule can be translated into a protein that actually does things for your body for the most part. Right? DNA is only the blueprint, the instruction on how to do a certain task, but in order to actually do the task, it's the protein that comes in place. So, so we have to talk about the process of gene expression, meaning that how do we go from the DNA that you start with, the instruction, into the application, which is the protein. So this is the central dogma. Is DNA transcribed into RNA? This is a transcription process, and RNA is translated into protein. So this first part, DNA to RNA, is called transcription. RNA to protein is called translation. And then the gene expression, it's the same idea as central dogma, is that the expression of a gene, and the gene is a sequence of DNA that codes for a protein, DNA coded into protein, right? So there's a difference between somatic cell and gametes, and I hope by this point you know what they are. We talk about it probably more than 20 times in class. So we have all kinds of cells in our body, actually hundreds of different kinds of cells in our body, about 200 actually. Somatic cells are any kind of cell that is not a sperm or an egg. Okay, a somatic cell. So let's see. Let's a quick review on somatic cell. We did this in class today already, but somatic cell review again. So somatic cell is not a sperm and egg cell, right? And they're also always diploid, meaning that it's 2N, meaning that there are homologous chromosomes. And if you don't know what homologous chromosomes are, I'm just really sad. But if you don't remember what homologous chromosomes are, they're chromosome pairs that looks like this. Let me find you a picture. See what I'm doing, right? I can see this picture in my head, but I can't tell you what it is. Sometimes you might not know something, and instead of just being like, I don't know what it is, Google it. Google has everything. Might not be completely correct all the time, but it's helpful, okay? So if you look right here, for example, this is a good example of a homologous chromosome. This is one pair of homologous chromosome. This is one pair of homologous chromosome. They're both duplicated chromosomes. So let's see. Let's get a karyotype. So if we look at a karyotype of a normal, this is a normal human, this is better, a normal human male, right? You see these, for each type of chromosome, there are 2 chromosomes that can pair up. They can pair up during meiosis 1 because they code for the same thing. So if you were to have a protein right here that codes for the color of your toe, that's not a thing, I just made it up. But this right here will also code for the color of your toe, okay? So the genes on this one chromosome, one homologous chromosome, is always the same as the genes on the other one. And then you wouldn't have color of your toe right here, for the most part. So those are homologous chromosomes. And this right here is a look of a somatic cell, because somatic cell is diploid, meaning that there are homologous chromosomes existing. So non-sperm egg, diploid cell, their homologous chromosomes replicate through mitosis, right? So remember, when we go through the process of mitosis, you make the same cell. So if you were to produce more skin cells, mitosis will allow that. And then there are gametes. So those are sperms and eggs. And they're haploid. And they're 1N, and there's no homologous chromosome. No, gotta make no homologous chromosomes. Okay, so these are things that you have to understand, right? Before we can even talk about what is the effect of a mutation that happened to a somatic cell. What are the effects of mutations happening to gametes? You first need to know what are somatic cells, what are gametes, right? Again, one more that we missed. So in a somatic cell, there are 46 chromosomes, or you can call it's the same idea, 23 pairs of chromosome for a human. All right, so this is not all organisms, it's just human. Because gametes are haploid, or 1N, right? If 2N equals to 46, meaning that a diploid cell has 46 chromosomes, then this 1N cell is going to have 23 chromosomes. Or, well, they're just 23 individual chromosomes, right? You can't, there's no pairs anymore. If you just cut, where'd it go? If we just cut all these numbers by half, you'll get a human gamete. So let's see a karyotype of a gamete. See, right here, we don't have pairs anymore. We only have individual chromosomes. See how this one right here, this is different from what we have right here. They're pairs and no pairs. Haploid and, oh, there it is, diploid. All right, somatic cell, the other one's gamete. All right, so since we're talking about mutation, and there's a difference between mutations, somatic cells, and gametes, well, what are the differences? So if a mutation happens to your somatic cell, this mutation is not going to pass on to the offspring at all, right? And that makes sense because it's a part of your body and you're only producing gametes, you're only producing offspring using gametes. It's impossible, at least naturally, to use your skin cell and get another child, like right here, a copy of Miss Bai. That is impossible. That'd be very freaky. So that's the point, right? If there is a mutation in the somatic cell, it's only going to affect your somatic cell. It's not going to affect the offspring. However, if there are mutations happening to the gametes that are produced, and it just so happened that this particular gamete, by chance, is the one that's fertilized and become the offspring, and since the entire offspring comes from this one original cell, every single cell of the offspring's body is going to be affected by this one mutation that happened to your gamete. That this is it sounds kind of scary. It is kind of scary, actually. But This is what happens. This is how people become very sick, you know, or might not even be able to live as long as you do because of a mutation that just, you know, randomly happens to a particular gamete. It doesn't even have to be every single gamete, but if it happens to one gamete, and it just so happened that this one gamete becomes fertilized and become a child, this child can potentially be very, very sick. Or it might not. So that comes to our next question that I forgot to add on here. What are the effects of a mutation, right? There are ones that are beneficial. There are ones that are that have no effect. Oh, and there are ones that are harmful. That's the three possible effects. When we think about mutation, you shouldn't just think mutations are bad for you. That is not necessarily true, right? The existence of human being is because of mutations. The existence of this cat just got spooked. Hi, cat. The existence of this cat, and what makes this cat different from your cat or other cats, is because of the mutations. The reason why I look different from you, well, there are many reasons, but even if we were to be the same gender, I will look very different from you because there are mutations, right? So that's not necessarily a bad thing, and it actually allows you know, populations to survive better in changing environments, and the environment is also changing. And so there's beneficial. A lot of the times a mutation can have no effect for two reasons. One, when the RNA before an RNA is translated into a protein, there are parts of introns and exons. The introns are removed before the RNA is coded into protein. Okay, so the intron regions kind of get trashed and doesn't actually get made into protein. So if you were to have a mutation within the intron region, it's going to have no effect on the protein that's being made, right? Remember, if protein is doing the job and there's no effect, no difference in the protein, then everything is fine, or there's no effect, right? Another reason is that we have to look at a codon chart now. If we look at a codon chart, such as this one right here, then you can see that there's the redundancy in codons. Redundancy meaning there are the repeats in codons, right? CUU is not the exact same RNA, mRNA codon as CUC, but it codes for the same amino acid. So if there's a mutation that changes CUU into CUC, it's not going to have an effect on the amino acid produced, and it's not going to have an effect on the protein that's produced as well. Okay, so now we're on to point mutation and substitution, and the differences between all of those, which comes to this. Oh, you can't see it actually. Which brings you to this chart that you didn't get, but now you get to look at it. You don't get to use it because you didn't show up for the review, but that's okay. Is it okay though? Anyway, so we have this chart that I made going over all the different types of mutations. So if you take a look at the overview of this chart, it shows you that mutation is our center word, right? And there are two big categories. One is called point mutation / substitution. One is called chromosomal mutation. Point mutation means there's only a change in one DNA nucleotide. So if you look at this original DNA right here, and you compare it with this mutated DNA right here, there's only change in one base, such as this one, C. We changed A into a C, right? Change in DNA nucleotide. Or you could have this. Instead of changing the last, right here, this is our original, and then we have this mutated, and we changed this one C. We swap it out. We didn't add, we didn't lose, we swapped it, okay? We got rid of the C, and then we added the A. We did a swap, okay? This is called a point mutation. Again, right here, as you can see, there's a change in one DNA base. This is also a point mutation. But you know, the whole idea of mutation is we start with DNA, we end with protein, and we're asking ourselves, is our protein going to function just fine? But you have to start from DNA, right? So when you look at a codon chart that I give you, it's going to be an RNA codon, so RNA to amino acid. So if I were to give you a DNA sequence, you have to go through the transcription and translation process before you're able to know whether there's a change in amino acid or not, okay? Well, if you need me to repeat, just go back and listen again. All right, so now we're going to focus, so these are the two big categories, point mutation and chromosomal mutation. This one big category has three subcategories called silent, missense, and nonsense mutation. The point mutation, see, it changes one DNA nucleotide. If you change one DNA nucleotide, it's all obviously going to change one RNA nucleotide, and that's going to affect a codon in different ways. Silence, missense, and nonsense, they're different in the way that they have different effects as a result of the point mutation. So there are three different results of the point mutation, okay? So silent mutation always means that even though there's a change in the DNA, it doesn't affect the amino acid coded by that corresponding RNA codon at all, which means the protein is going to fold just fine and there's no effect of the mutation, right? So let's take a look at this example that I gave you guys. Well, all right, so we have silent mutation on the top, changing one DNA nucleotide. Original DNA, see, I changed this A base right here into a C right here. So the corresponding RNA, it was supposed to be GGU. That's before the mutation, but after the mutation, now it's GGG. So if we were to look at our codon chart, GGG codes for glycine, but the original GGU also supposed to code for glycine. See? Even though there is a point mutation, there is no change in the amino acid at all, like glycine and glycine. And that we call it a silent mutation because it's a point mutation that is silent, it's not changing anything. Okay? The next one is called a missense mutation. A missense mutation does change the amino acid to another amino acid. So we're not taking out amino acid, we're not putting the extra amino acid, we're changing it. So if you look at this example right here, we started with glycine. That's the original amino acid before the mutation, but after the mutation, we got a valine as a result of this, changing the C to A, which causes the RNA to change from GGU, where this one codon GGU is changing to GUU. A codon is 3 bases of RNA sequence that you read, well, that the ribosome reads. All right, so then if you look at the original amino acid sequence versus the mutated amino acid sequence, there's a change. All right, so anytime you have a change in the amino acid, if we change leucine into proline, changing proline into threonine, whatever, anytime you change an original amino acid to a different amino acid as a result of the mutation, we call that a missense mutation. The last one is called a nonsense mutation. There are only 3 possible ways because a nonsense mutation changes the original amino acid into a stop codon. So for example, right here, you're supposed to have these 3 amino acids coded by the original RNA. However, due to this one mutation, changing this G base, DNA base, into a T and changing the C into A, instead of having an amino acid right here at all, we got a stop codon, which means translation will stop, right? So let's say you're supposed to have a protein that has 15 amino acids and there's a mutation, a nonsense mutation happening to the 4th codon. Now your amino acid sequence is only going to be 3 amino acid long. And so instead of having a full length protein that can fold into an actual protein and work, you only have this tiny bit, and this is not going to work. This is called a nonsense mutation. So if you're comparing these 3 different types of mutation, nonsense is the worst. Okay, let's see if we answered all the questions. Three types, now you know what they are. You know the differences. You know the effects. How are they different from the chromosomal mutation? I'll talk about that. Okay, so there are many ways that I can ask you these questions. I can give you, I can ask you, what is a silent mutation? And I could have a list of 4 different descriptions that you can pick one out for me that is the most accurate silent mutation definition. I can give you a DNA sequence before the mutation and DNA sequence after the mutation, and you have to go through transcription and then translation and see what is the difference in the amino acid sequence. If there's a change in amino acid sequence by 1 amino acid, such as this one, you will know that this is a missense mutation, right? So make sure you first, when I give you a sequence, if I don't tell you whether it's DNA or RNA, you should think to yourself, does it have T in there? Does it have U in there? Because if it does have T in there, that has to be in DNA. So you have to go from DNA, RNA, protein, amino acid. If there is a U base in there, then you have to go from RNA to amino acid. Okay? It's not too difficult, I suppose. Now, chromosomal mutation has more effect. Chromosomal mutation affects a larger part of the chromosome. We're not just changing one nucleotide, and we're not just affecting one amino acid. Chromosomal mutation can affect a lot of amino acids within the one protein that's being produced. Now, there are 4 big categories of chromosomal mutations, and within the first category, there are 2 subcategories. So if you look at this chart, it's very visual, in my opinion, on the different types of mutations. So the first one is called frame shift mutation, because the reading frame shifts. Okay, and we're going to take a look at how that works. There's duplication, there's inversion, and there's translocation. So the first one, frame shift mutation, happens when there's either an insertion or a deletion. An insertion or deletion doesn't change the chromosome number, okay? So if your cell is supposed to have 46 chromosomes or 23 chromosomes or however many chromosomes there are, an insertion or deletion does not change that chromosome number. You will still have 23 or whatever chromosomes, okay? An insertion happens within the chromosome. So if you were to have this pen right here, okay, an insertion happens like this. Sorry. This is still one pen, but now it got an extra piece in the middle. See the difference? You remember how a pen's supposed to look like, and it doesn't look like that anymore? This is insertion, okay? We insert an extra piece. It could be 1 base. 1 nucleotide, it could be 2, it could be 3, it could be 20, it could be 500. It doesn't matter as long as you're inserting an extra piece of, an extra part of chromosome within the original part, you call that insertion, okay? That's insertion. Oh God. A deletion is you delete a part. It's hard to show you right here, but I guess this works. We have a pen right here. We lose a part. That's deletion, okay? How would it look like on DNA, because I'm going to give you some DNA examples and you're going to identify for me what kind of mutation it is. So if you look at this example that I gave you, we have the original DNA sequence and we have the mutated DNA sequence. What I did here is I added an extra C. How would you know that I added a piece? First off, you can look at the overall length of the DNA. Right here we have 9 DNA bases in total, and right here we have 10. So if you think about it, something happened, made it longer, right? Remember this? I'm doing it again, just for you. See how this chromosome got longer? Normally, if you have a chromosome that got longer, it's probably insertion, but just to verify it, you want to take a look at the overall bases, right? You want to see the last base is still C. The second to last is A. This is fine. The third to last is G. The fourth to last is supposed to be A, and that's all right. The next one is supposed to be C. The next one again is supposed to be a C again. And then the next one is supposed to be a C. But oh wait, the next one's supposed to be an A, which is right here. How do we have 4 C's in a row instead of only 3 C's in a row? Ta-da, there must be an insertion mutation that inserted an extra piece, right? You're supposed to have 9 bases, now you got 10 insertion. Okay. So now, what is the effect of this? If you were to compare the RNA sequence, obviously the RNA is also going to be longer. But here's the magic part. This is called frame shift mutation because the reading frame is shifted. Okay? You're supposed to read it as tach CCA GAC. But now you got tach CCC AGA C. So the entire reading frame shifted. And I'm going to show you in real life how this works. You ready? Ready for the magic? See, this is my original sequence, right? Now I'm putting in an extra C. Now we got an extra C, but we still have to read 3 bases at a time. How do we do that? Now we have to read CCC, right? So everything else have to shift by 1. The C right here supposed to be right here, right? If I want to add this extra C, see this C is going to shift to the right. Oh, no, I totally messed up. Wait. See? Everything shifted to the right. You can see it again. See, everything shifted, right? But when everything shifts, and because your DNA doesn't have these natural, or RNA, they don't have those natural spaces in between. You know we put spaces in between because that's how we read a codon. So you're going to do CCC, then now this one has to move. Now you have to group these 3 letters together, and this one is left over. Here we go. I got to make this one right again. All right, so that's what we call a frame shift mutation. Because you're shifting the frames. You're shifting the reading frames, the reading of the codon. Okay? Okay. All right, now you want to see the effect of this mutation. The effect of this mutation is the amino acids, every single amino acid after the mutation is changed. Okay, so in this case, we have these 3 amino acids, but you see we're not just affecting this one amino acid that's affected by the second codon, which is the direct effect of the insertion. You can also see that the 3rd codon is also changed. Before we were to have a 4th and 5th, whatever, all of those codons are going to be changed, so all of the amino acids are going to be changed. Same idea with deletion, right? This is our original DNA sequence. This is our new DNA sequence. So let's see. Let's see it in real life. If I were to delete this T, now everything shifted forward, but we still have to read 3 bases at a time, right? So now that you read 3 bases at a time, you get this mutated DNA sequence, right? So that's what we call deletion, and this also causes a frame shift mutation because the reading frame for the codons are changed. The result is every single amino acid after that change, that mutation is going to be changed as well. You're going to see one again, insertion mutation. We added an extra A. If you cover that A, you can see everything else after this is the same as what it's supposed to be, but every single amino acid is changed to what it wasn't supposed to be. Same thing with deletion. All we did was to get rid of the C, but now instead of reading CAT, TCA, CAC, we're going to read ATT, CAC, ACG. It's all wrong now. So every single amino acid after insertion or deletion or any kind of frame shift mutation, which is what insertion and deletion are, every amino acid is going to be changed. Okay? Now, the next one is called duplication. Duplication, you take one part of the chromosome, you duplicate it. So let's pretend that this is the same kind. You make a duplicate, right? So how would you know that it's a duplicate, not an insertion? You see if it's the same as a part, it's the same as a part as what it was before. So for example, if we have this DNA right here, and you see that there are some extra bases, but they're not just some extra random bases, they're extra repeated sequence of these 2 codons, that has to be a duplication because you're duplicating, okay? So as you can see, the effect on the amino acid sequence is you're duplicating part of the amino acid as well. But this is nice and clean. We're duplicating 2 complete, mRNA codons right here, caused by change in a duplication of 6. Of 6 nucleotides, but this doesn't have to happen. It's also possible that we'd only copy 5, and this is not going to give you a clean, glycine, leucine, glycine, leucine. Again, this last one is not going to be leucine. I don't know if you get it. I hope you got it. Okay, this is a clear cut because it's a, you know, it's 3. If you're making copies of 3 or 6 or 9 codons, then you'll get amino acid sequences that are just the repeat of part of the original. But if you do like 2, for example, if you make this CC, it's not gonna look like this, right? You don't just have a repeat, and you're not just gonna have a repeat of the amino acid sequence. You're gonna have some different amino acids right here. I hope you got it. Now we're getting there, guys. We only have 2 more. Inversion is when you take a part of the chromosome and you flip it. So let's say you have this right here. You flip it. That's inversion. That's it. How would you supposed to be? How would you know? You look at the original sequence. You look at the new sequence, and you try to find differences between the two. Right? It's like you're finding clues. So you have this DNA. You have this original DNA, you have this mutated DNA, you see that there's no change in the length of the DNA, so it can't be frame shift or duplication because there's no change, you didn't add any bases. So now you want to see if there's, anything that got flipped. And you see that there was TAC and now you got CAT. There must be things that are flipped. Here we go. But again, you don't have to just flip 3 or 6 nucleotides. You could do 4 as well, and it's going to mess things up even more. So right here, as you can see, we got a different amino acid because we flipped things, and flipping nucleotides is not going to, give you the same amino acid. All right, we're last one. Whoa. Sorry, that's my mom. Okay, the last one is translocation, I think. Oh, it's not. Translocation. Okay, translocation. We got these 2 different chromosomes. Every single mutation that we've talked about so far are single chromosomes and weird things that would happen to a single chromosome because accidents happen in your body. But translocation involves 2 chromosomes always. And what happens is, just like crossing over, translocation will allow the 2 chromosomes to swap apart. And how are you supposed to know that they swap apart? You look at what I give you. I'm going to give you 2 original DNA instead of 1. And you're going to see that part of the sequence got switched. So in this case, I switched these 3 with this first one. And now the amino acid sequence is also, there are parts that are switched. But it doesn't necessarily work out that nicely. It's also possible that only those 2 switch to this one with this one, and then the amino acids that are being coded are not going to be the same. Okay. So how can we represent this pretty easily? Deletion, you got to make it shorter. You delete a part. A, B, C, D, E. We got rid of D. A, B, C, E. Duplication, you duplicate it. A, B, C, B, C, D, E. Inversion, A, D, C, B, E. And then the last one, this is translocation, where you have E, F, G, M, N, O, P, and M, N, O switches with A, B. Translocation. All right, I know you're so done with this, and me too. I think we're done. Okay, you should be able to write out an example as well, just like this. You either add or delete or duplicate or you invert or you translocate, you swap. Okay. Next video we'll go over karyotyping a little bit, and then you will be set for the test.